We'll be doing another bioconcept in this video, which is Lac Operon, and how it functions as a regulating unit in certain prokaryotes in response to the environment. So the contents of the video will be the introduction to the structure and function of the Lac Operon and how it functions in terms of a molecular aspect and in terms of a biochemical aspect. As we already know from the molecular biology, the gene expression of each of the cell is actually in response to the conditions in which the cell is existing. So the genetic material which each cell contains, which is required for its growth and development. However, the genes which contain all the kinds of an information are needed to express at certain times. So all these genes which are needed for their day-to-day functioning and maintaining the regulation of all the vital biochemical processes like respiration, metabolism, these genes are needed to be expressed at all times. While as there are certain kinds of the genes which are needed for the regulation or for the expression for certain times or at a particular point of a life of a cell. So these genes need to be expressed only once or few times, and most of the times they need to be switched off. This regulation of expression is done in a different kind of manners. One of the important is the heterochromatization and methylation, as well as acetylation in eukaryotes particularly. However, in prokaryotes, the gene expression is regulated with the help of a specialized units of structures, which is ahead of the expression part of the gene. This concept for the first time by Jacob Monod and Loaf in 1961. This concept was called as the operon concept. What is an operon? It is actually a group of genes which are transcribed together at the same time. And this operon is actually a polycistronic kind of a gene which produces a number of different kinds of enzymes or proteins during this its expression. And this operon is expressed in response to the environment in which the cell exists. So it usually is found in prokaryotes and usually controls the important biochemical processes. As you can see on the right hand side of the picture here, this is the general concept of the operon. It consists of certain regulatory genes, the promoter and operator region, and the actual genes which are to be expressed, which produce a number of different kinds of proteins, which in turn regulate the biochemical process in prokaryotes. So hence this whole operon concept is actually the expression or the regulation of the expression of some genes in response to the environment, which mediates its expression or not. Although there are a number of different kinds of operons which exist in the prokaryotic world, one of the classical examples can be cited from Lac Operon. Lac Operon consists of three functional units. First unit is the structural gene in itself, which is called as LAC-ZYA. This LAC-ZYA is here, the structural part of it, which in turn code for three different kinds of proteins. Then there is a upstream regions which are operator and promoter sequences called as the LAC-O and LAC-P. As you can see here, LAC-O and LAC-P similar to that of promoter and operator region, to which the RNA polymerase need to bind and identify first, and then it needs to clear it from the promoter region to go into the expressive structural region to express this part of a Lac Operon, and finally to synthesize the three different kinds of proteins which are needed for the regulation purpose. And then there is another part of this operon which is called as the regulatory sequence. The regulatory sequence in Lac Operon is LAC1 or LAC-I. LAC-I stands for inhibitor of the LAC. it expresses certain protein which acts as an inhibitor of the expression of these LAC-ZYA genes. Lac Operon, as I said, that there are three structural genes which are coded as polycistronic mRNA. That is, it is expressed as a single mRNA molecule, out of which three different proteins are then finally synthesized. These three different proteins or polypeptides are the beta-galactosidase enzyme, which hydrolyzes the lactose and converts it into or degrades or breaks it into two different monosaccharide units, that is glucose and galactose. Another one is permease. This is an important protein which functions by inserting itself into the cell membrane and enabling the transport of the lactose from the outside ambient environment into the cell, so that this lactose can then be utilized by beta glucosidase and generate glucose, which in turn can be metabolized for the existence of the cell. Then third is called as the transestalized, but what it is function is nobody knows so far. As I already told you that these operon models function in adapting the cell to its environment, to the source of the energy. For E. coli, the sources of energy can either be glucose or lactose. However, as we know that lactose, when it is metabolized, it has to first undergo hydrolysis by galactosidases to generate glucose and galactose, and then it can be later on degraded metabolically. So E. coli has a preference for glucose only. When glucose is present, it doesn't go for the lactose. But when environment forces the E. coli cell to work or to feed on the lactose in absence of glucose, it is able to do so. So generally bacterium, this bacteria usually prefers the glucose whenever it is possible from the environment. If however it is not available, then lactose is utilized for the energy generation. The regulation of these structural genes and their expression in the environment depends actually upon the availability of the carbohydrate. If glucose is present or not, or if lactose is present or not. So this model, which is a Lac Operon model, is actually the utilization of the lactose by the E. coli cells. And we can discuss this in two different ways. There is a molecular aspect of it and there is a biochemical aspect of it. So focusing first on the molecular aspect of the regulation of Lac Operon, we now so far we know that Lac Operon contains the structural gene, promoter, operator, as well as a regulator gene. Regulator gene here is a Lac-I. Lac-I, when expressed, it expresses as mRNA and then is translated into a protein. This protein is functional as dimer, two subunits of the same protein. When it is functional, it goes and binds to the operator region of this operon. By binding to the operator region, it prevents the RNA polymerase from promoter clearance and hence movement and expression of the LAC-Z by a structural gene. So when repressor is bound to the operator, the RNA polymerase will not be able to move from the promoter region and it will not be able to express the structural ZYA genes, and hence there will be no three different proteins expressed. When does it happen? It happens when lactose is totally absent in the environment. In turn, it also means that glucose is present. So E. coli is usually utilize the glucose then, and it doesn't need the expression of beta glucosidase or permease to utilize the lactose because lactose is not present. So when lactose is absent and glucose is present as high concentration and available as a sole carbohydrate, then the LAC-ZYA genes are not expressed, and we will find the Lac Operon be repressed by the repressor, which is expressed by the regulator gene. This is the first kind of a molecular aspect of the regulation. Another aspect of molecular regulation is through CAP. CAP is a CAMP activating protein. cap binds to the promoter region a little ahead of the RNA polymerase. RNA polymerase needs it for the identification of the promoter much avidly than when it is not present. So what does cap do? Cap actually binds to this promoter region. It produces a 90-degree kink in this promoter region, and by doing it in this way, it enhances the binding of an RNA polymerase to the promoter, and then also enhances the promoter clearance from the promoter towards this structural gene, and hence increases the expression of this Lac Operon structural genes, producing more and more beta glucosidase and permeases. When does this happen? This happens when there is a complete absence of glucose, or glucose is very low in concentration, and concomitantly lactose is very high. So what happens that when lactose is high, we need certain enzymes, these two kinds of enzymes, these beta glucosidases and the permeases, to utilize the glucose as the sole source of energy, and hence we need the expression of these structural genes of the Lac Operon, and this causes the cap activation of the promoter, in turn binds the RNA polymerase much avidly, and hence the expression will be enhanced, increased from the basal levels to produce more and more of the enzymes. So you can see in the following diagram, it is quite expressed in terms of the low glucose and the high glucose. When there is a high glucose, cap will not be able to bind to the cap site, and hence the promoter clearance of the RNA polymerase will be very low, and hence the low transcription and low structural expression of these genes. Whereas when there will be a low glucose, or glucose is very much not present and lactose is to be utilized, then there will be a high expression and high transcription of the structural genes, so that the more and more lactose will be uptaken and then degraded by galactosidase to feed it to the metabolic pathway for utilization. So comparing the molecular aspect, when glucose is low or absent, what happens in the E. coli cell is that CAMP levels are very high, and in turn the CAMP will dimerase the catabolite activating protein cap, and then cap will attach to the promoter, as you have already seen in the previous slide. By attaching to the promoter, it will cause the enhanced activity of the RNA polymerase by it is identifying and then quickly binding to the promoter region, and then facilitating the promoter clearance quickly, and hence the expression of the structural part of the Lac Operon will be enhanced, and we will have more and more of beta galactosidase and more of permeases, which will allow the E. coli cell to utilize the lactose preferentially, which is available there, while as the glucose is low or absent. And in contrast, when lactose is low or absent, what will happen is that the Lac-I will be synthesized. Lac-I will cause the repressor to dimerize and then attach to the operator. When it binds to the operator, the RNA polymerase will not be able to do the promoter clearance, and it will not be able to express the Lac-ZYA gene structural part of the operon, and hence the products that beta galactosidase and the permease will be very much low in the E. coli, and we will be able to use this E. coli will be able to use only glucose. So this also happens when lactose is present. So in turn we will say also when glucose is absent, lactose is present, and this model will happen when lactose is low or absent and glucose is present. So this is the molecular aspect of it. Focusing on the metabolic aspect of the regulation of the Lac Operon, and hence the expression of the structural unit of this operon, this depends upon the relative availability of the glucose and lactose for the E. coli cell. And there are usually four different kinds of scenarios that can be present in the environment where the E. coli is living. If glucose is present and lactose is absent, then we don't need the structural genes to be expressed, and hence E. coli does not produce beta galactosidase gene. Similarly, the beta galactosidase gene is not expressed when both glucose and lactose are present. And third scenario is also the same kind of a scenario when both glucose and lactose are absent. When both are absent, we don't need to utilize anything. Then only one scenario is there when the structural genes are expressed based upon the relativity of the glucose and lactose. That is when glucose is low or is absent from the environment, and lactose is present in high concentration so that it can be utilized by the E. coli cell for its metabolism, and hence the E. coli cell must produce beta galactosidase at this kind of a scenario for lactose utilization. And we know from the molecular biology what happens in this scenario is that the polymerase which is bound to the promoter region will be able to clear the promoter region and hence express the LAC-ZYA genes. Furthermore, also there is the upregulation by the cap proteins. In the first scenario, when lactose is absent, what happens is that repressor protein, as we already know from the molecular aspect, is that the repressor protein will be continuously synthesized by the LAC-I gene. It will be able to dimerize, and as a dimer, it will bind to the operator gene, preventing the expression. And the promoter site, even though it will be able to settle the RNA polymerase there, but RNA polymerase will not be able to clear the promoter. Promoter clearance will not happen when lactose is. In the second scenario is that when lactose is present, then what happens that lactose, whatever present in the ambient condition, will be uptaken by the basal activity of the permease. When it enters the cell, it will be converted to allolactose. Allolactose will fit into the repressor dimer protein, and it will change its conformation, change its structure so that now it is inactive. When it is inactive, it will not bind to the promoter region. When it will not bind to the promoter region, the RNA polymerase which is already there in the region will be able to clear the promoter and hence express the structural genes, producing more and more of permease and beta galactosidase. And permease will bring more and more lactose from the ambient environment into the cell, which will be then degraded, hydrolyzed by the galactosidase and hence utilized. So this happens when lactose is present, but there is a condition also that glucose should be absent at this. So up to this point, we know that the lactose, if present or absent, will drive the ability of the regulation of the Lac Operon, specifically for the expression of the genes LAC-ZYA, only when lactose is present as a sole source of carbon or as a sole source of energy. What will happen for the E. coli cell which is living in the environment in which glucose as well as lactose is present at the same time? Albeit also when glucose is high, even though lactose is present, what will happen in those kinds of scenarios? What happens is actually the RNA polymerase is already bound to the promoter region, but we know from the molecular aspect we have covered it that RNA polymerase is able to express the gene at a very low expression because it usually is not able to bind and identify the promoter region with much avidity. For that to happen, it needs a cap protein to bind. So when lactose is present, even though the glucose is high, so at these conditions, the E. coli cell is able to utilize glucose preferentially. Why? Because the RNA polymerase, even though bound to this promoter region of the Lac Operon, it is not able to move and do the promoter clearance with high fidelity, and hence there will be a low expression and low availability of the enzymes which will utilize the lactose. And in these conditions, when both are present, glucose is preferentially utilized. Why? Because of the RNA polymerase's inability to identify the promoter with much high fidelity, and also to move from the promoter region to the structural region to express it. So the sole scenario when lactose is present and glucose is either absent or low, we need a catabolite activated protein to actually enable RNA polymerase not only to stabilize at the promoter region but also to identify it first much avidly, and then cause the promoter clearance much quickly from the promoter towards the structural genes to express them. So when there is a low glucose, there is a high CAMP concentration. This CAMP in turn causes the CAP proteins to be dimerized and activated, which in turn will bind to the cap site ahead of the promoter of the RNA polymerase, which will introduce the kink and enable the RNA polymerase to go through the high transcription of the structural genes. So in a scenario where the glucose is low or is totally absent, the CAMP is available for the CAP to function, and the catabolite activator protein will function under these conditions to enable the E. coli cell to utilize lactose preferentially. Overall, four scenarios of the absence or presence of the glucose versus lactose can be shown in this diagram, and you will see only one once when glucose is not present and lactose is present to be utilized, the activator protein CAP will bind to the DNA. The repressor protein will be lifted off the operator site, and the RNA polymerase is already present at the site of the promoter region, and then it will be forced to do the promoter clearance with much high frequency, leading to the transcription of the Lac Operon at a high concentration, and hence enabling the cell to utilize lactose preferentially when there is no glucose present. So this was essentially everything about the Lac Operon and its structure and function, and how it regulates the utilization of the lactose during the glucose absence in the E. coli cells. If you like the lecture, do visit my Amazon page for my books. They are available both in Kindle and paperback format. You can order them for your utilization. And do subscribe to my channel for further videos which I will upload in due time.